Good day, this is Professor Will, CBMD, PhD. Today is Monday, January 13th, 2020. It is 7:36 p.m. Eastern Standard Time. Today's topic, transformation genetic. So let's get started here. Genetic transformation is a process by which bacteria and microbial eukaryotes take up fragments of negative DNA from the extracellular medium to produce genetically different progeny cells called transformants. The process is manifested by genetically ecologically diverse organisms in which it is encoded by chromosomal genes. Natural transformation systems require the development of competence by the recipient cell as a specialized physiological state necessary for binding and uptake of exogenous DNA as linear fragments. Considerable diversity exists among the transformation apparatus of various bacteria, with some showing specificity for homologous DNA, but a unifying theme of natural system is the uptake of random fragments of single-stranded ssDNA. Transforming DNA that is related at the nucleotide sequence level to the chromosome of the recipient cell can be incorporated into the residence genome of homologous recombination to give a region of heteroduplex DNA. Mismatched bases may block formation of the heteroduplex or be removed from the recombinant structure by excision from the donor strand. Plasmid DNA is also taken up by natural systems as single-stranded fragments. These may interact to regenerate circular plasmid molecules, albeit with low efficiency. Natural transformation is viewed as a gene exchange process that enhances the genetic diversity of bacterial population, as well as maintaining the constancy of bacterial genome by potentiating recombinational repair of damaged DNA. Artificial transformation procedures have been devised for many microbial species, allowing the uptake of duplex and circular forms of DNA. Such procedures provide a cornerstone of recombinant DNA technology. Natural transformation is encoded by genes in the bacterial genome and involves the uptake of negative DNA from the extracellular environment. Hence, there is no requirement for a living donor cell. The process supports the uptake of random small fragments of DNA. These are rendered single-stranded during their transport into the recipient cell and are recoverable as material of about 10 to the power 4 nucleotides in size. Transformation differs significantly from the two other quasi-sexual processes for gene transfer between bacteria, namely bacterial conjugation and transformation. Transduction is a bacteriophage-mediated process. It involves the accidental packaging of a double-stranded fragment of bacterial or plasmid DNA of the donor cell into a packaged particle, which then acts as the transfer vector to the recipient cell. The size of a transduced DNA fragment is generally greater than that of transforming DNA. It may exceed 100 kb depending on the DNA packaging capacity of the phage particle. Bacterial conjugation is a dedicated transfer process of self-transmissible plasmids in a class of mobile genetic elements called conjugated transposons. According to the general model of conjugation based on systems studied in Escherichia coli, the process requires a specialized contact between the donor and recipient bacterium to form the transport pore for a specific DNA strand. This is transferred progressively in the 5-transposon, 3-transposon direction from a genetically defined origin. As a defined property, conjugated plasmids can cause the transfer of very large sectors of chromosomal DNA, which may exceed 2000 kb in size. Many genetically and ecologically distinct bacteria are known to be naturally transformable. The list includes gram-positive and gram-negative EU bacteria, including cyanobacteria and archaea. Mechanistic studies of transformation have focused on the similar systems determined by S. pneumonia and the soiled bacteria Bacillus subtilis. These are viewed as gram-positive models. Gram-negative paradigms are the systems specified by Haemophilus influenza and Neisseria gonorrhea, the etiological agents of spinal meningitis and gonorrhea respectively. The Haemophilus and Neisseria systems differ significantly from each other and from the gram-positive models. Transformation is likely to contribute importantly to gene flux between bacteria and natural environment. The process has also been used for mapping the relative positions of chromosomal genes of some naturally competent bacteria. Mapping involves the selection of transformants containing a specific donor gene and scoring the fraction containing one or more unselected donor markers. The derived values allow the ordering of genes on the basis that co-transformation frequency is inversely proportional to genetic distance. The usefulness of the technique is limited by the small fraction of the genome that is transferred on any one DNA fragment and by fastidious growth requirements of a number of transformable bacteria, which make it difficult to isolate mutations in sufficient density per unit of chromosomal DNA. Such problems are bypassed by recombinant DNA technologies that allow mapping with reference to restriction fragment maps, or in the case of B. subtilis and H. influenza, to the known nucleotide sequence of the entire chromosome. The need in molecular biology to introduce DNA modified in vitro back into cells has led to the development of artificial procedures for the transformation of prokaryotic and eukaryotic microorganisms that do not develop natural competence. Methods used to promote the uptake of DNA include the treatment of whole cells with metal cations and conjugation with a heat shock, use of brief high-voltage electrical discharge, and polyethylene glycol PEG treatment of osmotically stabilized protoplasts. Transformation systems require the acquisition of competence in physiological state, enabling the bacteria to take up high molecular weight fragments of exogenous DNA. In many systems, competence is a transient condition associated with the induced synthesis of a small number of specific proteins. Growth conditions favoring the development of competence by different bacteria varying considerably. For example, competence may occur throughout the exponential growth phase and decline at the stationary phase, as found for Deinococcus, radiodurans, and Mycobacterium smegmatis, whereas the process develops in the early to mid-exponential phase in S. pneumonia and at the entry to the stationary phase in B. subtilis. Competence is observed to be a constitutive property in N. gonorrhea, yet another variation is apparent in the fraction of cells that acquires competence under optimal inducing conditions. Competence development has been characterized most extensively for B. subtilis. Here it is post-exponential response involving a complex network of signals that sense the nutritional state of the environment and cell density. Many of the regulatory proteins in the cascade participate in other post-exponential phase responses such as sporulation, motility, and the production of extracellular degradation enzymes and antibiotics. Competence optimally develops late in exponential growth in a glucose minimal medium, or early stationary phase, but occurs in only a subfraction of cells, making up 10% or less of the population. The state is associated with a cell type that is more buoyant and relatively dormant in macromolecular synthesis. 20 or more genes have been identified for development of competence, which collectively function in an elaborate complex of regulatory networks involving cell density sensing systems, protein, phosphorylation of protein-protein, and protein-DNA interactions. The various regulatory signals converge to control the expression and activation of the key competence transcription factor, COMK. This is required for the expression of late genes in the COMC, COME, COMF, and COMG loci, which determine the biosynthesis of the DNA binding and uptake apparatus. A particularly challenging aspect of gene transfer is the nature of the specialized channels that allow DNA as a polyanion to pass through the hydrophobic environment of cell membranes and enter the cytoplasm of the recipient cell. Considerable variation exists in the protein complexes making up such channels. As discussed later, some of the DNA transporters show intriguing relationships to systems dedicated to protein secretion. These include systems for the secretion of exoenzymes and for the biosynthesis of type 4 phyli. The later are extracellular appendages that mediate the adherence of virulent bacteria to host eukaryotic cells, as well as the flagellum-independent form of translocation across surface called twitching motility. A common feature of the transformation systems that have been examined at the molecular level is that DNA enters the cytoplasm of the recipient cell in single-stranded form. Considerable diversity exists between systems with regard to DNA uptake, apparatus, and whether or not it shows species specificity in using homologous DNA. In the B. subtilis and S. pneumonia paradigms, DNA transport across the single-cell membrane of the cell envelope occurs through a surface-exposed complex of proteins. The transformation of gram-negative bacteria is complicated by the fact that the DNA must traverse an outer membrane which characteristically contains an outer leaflet of lipopolysaccharide LPS, as well as cytoplasmic or inner membrane. In H. influenza, DNA uptake proceeds through a specialized membrane vessel, whereas passage of transforming DNA across the outer membrane of N. gonorrhea requires the type 4 pilots. The inhibitory effects of non-homologous competitor DNAs indicate that B. subtilis and S. pneumonia take up DNA without sequence specificity. The first stage in DNA uptake involves a non-covalent binding of double-stranded DNA to about 50 competence-specific receptor sites on the surface of the cell. Duplex DNA is bound much more efficiently than ssDNA, ssRNA, or dsRNA. Binding is initially loose in that the DNA is released by washes of high ionic strength or by competitor DNA. Loose binding is followed by tight binding in which the DNA is resistant to release by washing but remains exposed to the external medium, as shown by the susceptibility to shear and exogenous DNA. DNA then undergoes double-stranded cleavage by one or more surface-exposed endonuclease which may be localized at the binding sites. The size distribution of the resulting fragments is in the range of IV-5 kb. The uptake of DNA, defined by its acquisition of resistance to exogenous DNase, commences 1 to 2 min after binding, and occurs at a rate of 100 nt divided by S at 28 degrees Celsius. Uptake culminates in the entry of ssDNA into the cytoplasm of the recipient cell. The uptake channel is thought to be an aqueous pore in a surface-exposed complex of proteins. The complex includes an entry nucleus that potentiates intracellular transport of ssDNA fragments with the associated degradation of the second strand and the release of oligonucleotides into the medium. The entry nucleus of the S. pneumonia is the 30 kDa membrane-bound end-A endonucleus. The polarity of its activity is such that the non-hydrolyzed strand enters the cell with a leading 3' transpose terminus. Further evidence for a single-stranded intermediate comes from the eclipse phase. This is a transient stage in the transformation process in which DNA that has been taken up into a recipient cell, as defined by its resistance to extracellular DNase, cannot be isolated. In a form that transforms the second cell, the explanation is that entrant DNA is single-stranded in the eclipse phase and therefore an unsuitable substrate for uptake by a second cell. Another feature of the eclipse is the coating of the entrant strand with the ssDNA binding protein. The eclipse protein is thought to protect the DNA from nucleases and aid the next step in the transformation process, namely homologous recombination with the resident genome. Good day, this is Professor Will, CBMD, PhD. Today is Monday, January 13th, 2020. It's 9:01:23 p.m. Eastern Standard Time. Well, this concludes my reading on transformation genetics. Hope you enjoyed listening. Have a great day.